again to our channel Bioholic with your Friday educator Olivia. So today it's the time for the fifth part of photosynthesis. In this class I will make you understand the light reaction in the easiest way. But before starting, I want to make it clear that if you missed the previous parts, then go and watch those first. Otherwise, you will feel difficulties to understand the light reaction properly. Okay? So now, let's begin. The full photosynthesis process includes mainly two phases, light reaction and the dark reaction. Light reaction depends on light directly, but the dark reaction depends on the products of light reaction. But yes, though it is named as dark reaction, that doesn't mean that it occurs during night. It also occurs during daytime in exposure to sunlight, but it is called as dark reaction because it is dependent on light secondarily and not primarily, as it depends on the products of light reaction, and the light reaction is totally light dependent. Okay? In the first part, I described the structure of chloroplast, where I told you about the grana and stroma. So the light reaction occurs in the granum and the dark reaction occurs in the stroma. We know during storm the sky becomes dark, isn't it? Thus, you can remember that in stroma dark reaction occurs, and so in grana light reaction will take place. Clear? The grana consists of stacked disc-like structures that is known as thylakoids. Last Friday, we have learned about the photosystems, where we saw that the photosystems includes chlorophyll that are present in the membranes of thylakoids. This is the structure of photosystem that is present in the thylakoid membrane. This photosystem participates in light reaction. So this is considered as the exact location of light reaction. Okay, so light reaction is a light dependent reaction, and in this reaction solar energy is converted to chemical energy. That's why this reaction is also called as energy conversion reaction. And it involves light absorption, water splitting, oxygen release, and formation of high energy chemical intermediates that are ATP and NADPH. And the main goal of light reaction is to produce this ATP and NADPH. Last Friday we learned how photosystem works and how the antenna pigments helps in the transportation of light energy to the reaction center. In light reaction, there are two closely linked photosystems that is known as PS1 or P700 and PS2 or P680, but PS2 works first and PS1 works later. So when light reaches the photosystem 2, the light energy of appropriate wavelength is transferred to the reaction center. Then the electrons of the pair chlorophyll A molecules gets excited, and then it transfers the electron to its specific electron acceptor phosphatin. And now to compensate for the loss of electrons from the reaction center of PS2 or photosystem 2, water is split in the thylakoid lumen. This process is known as photolysis. By the water splitting, oxygen is liberated as byproduct and electron goes to replenish the PS2 reaction center, and the H+ ions get accumulated in the lumen. So here H2O or water molecule is acting as electron donor. The electrons that are lost from the PS2 then enter into an electron transport chain or ETC, where the electron from higher energy level comes to lower energy level along the concentration gradient. From the primary electron acceptor, it is transferred to the plastoquinone at first, then it is transferred to cytochrome complex, This time, while transferring, H plus from stroma enters the lumen through proton pump, helping to store more protons in the lumen. Then from cytochrome, the electron is transferred to the plastocyanin. And then from plastocyanin, the electrons finally reach the reaction center of PS1 or photosystem 1. Again in the photosystem 1, the chlorophyll A then gets excited and transfers the electron to its primary electron acceptor. And again, from here, the electrons are transferred to FD, that is ferredoxin, which is an iron-containing protein. Here, ferredoxin NADP plus reductase is present, which is used to reduce NADP plus to NADPH by the help of those electrons. To form the NADPH, here you can see that H plus molecules are necessary. What do you think? From where the H plus is coming? Actually, H plus ions that are stored in the lumen returns to the stroma along the proton gradient via the transmembrane enzyme ATP synthase. And then it participates in the NADPH formation. ATP synthase uses the passage of H plus ions to catalyze the synthesis of ATP. So thus, in light reaction, NADPH and ATP are formed. Clear? So as you see here, the change of energy, that is, the electron transfer pathway occurs during light reaction shows a up down up zigzag pattern. If we draw the electron transfer pattern between these two photosystems, at first there will be a vertical bar, then a diagonal bar, and then another vertical bar. So this pattern resembles as the word Z. That's why it is also referred as Z scheme. Here again the light reaction pathway is depicted, where you can see both the NADPH and ATP are synthesized in the stroma, as the FNR or ferredoxin NADP plus reductase is present in the outer surface of the thylakoid membrane at the side of the stroma. Okay. In this very light reaction, the addition of phosphate in the presence of light or the synthesizing of ATP by cells is known as photophosphorylation. And the photophosphorylations are of two types, the cyclic photophosphorylation and non-cyclic photophosphorylation. We already learned about the non-cyclic photophosphorylation, that includes the steps from the excitation phase of pH2 reaction center to the NADPH formation. But in case of cyclic photophosphorylation, there the pH2 or photosystem 2 is absent. When light is absorbed by photosystem 1 itself, the excited electron may enter into an electron transport chain to produce ATP. In this case, the de-energized electrons return to the photosystem back, restoring its electron supply. Hence here you can see a cyclic pattern. That's why it is called as cyclic photophosphorylation. And cyclic photophosphorylation does not produce NADPH, as NADP plus cannot reduced here, as the electron returns back to the photosystem 1. Now the differences between cyclic and non-cyclic photophosphorylation. So the cyclic photophosphorylation produces ATP and non-cyclic photophosphorylation produces ATP and NADPH both. And in case of cyclic photophosphorylation, oxygen does not take part. But in case of non-cyclic photophosphorylation, oxygen is produced by photolysis of water. In cyclic photophosphorylation, only photosystem 1 participates. But in non-cyclic photophosphorylation, we have seen the participation of both photosystem 1 and photosystem 2. In case of cyclic photophosphorylation, electrons released from PS1 goes back to the same source. No electrons from any other molecules enter in the system. But in case of non-cyclic photophosphorylation, electrons released from PS1 and PS2 does not revert to the main source. And outer electron that is released from the photolysis of water entered the system to replenish the photosystem 2 reaction center. Then the efficiency, the cyclic photophosphorylation usually occurs during anaerobic condition, when the light intensity is less and there is lack of carbon dioxide concentration. Non-cyclic photophosphorylation usually occurs during aerobic condition with high light intensity and high carbon dioxide concentration. And cyclic photophosphorylation usually occurs in bacteria-like organisms, but the non-cyclic photophosphorylation usually occurs in carbon dioxide fixative plants and other photosynthetic organisms. Okay? Now another important thing is the chemiosmotic hypothesis. Chemiosmosis is the biological process of producing ATP molecules through the action of ATP synthase. In 1961, Peter Dennis Mitchell theorized the chemiosmotic hypothesis, which explains how the energy molecules, that is the ATP, are created during photosynthesis. During lining the light reaction, we have noticed that ATP is produced as a result of proton gradient that exists across the thylakoid membrane. The components necessary for the chemiosmosis process are proton gradient, the enzyme ATP synthase, and proton pump. This is the schematical diagram of chemiosmosis, where due to water splitting, protons are liberated which are accumulated in lumen of thylakoids, and outside the thylakoid membrane, that is in stroma, protons are used to reduce NADP. So number of protons decreases in the stroma and number increases in lumen of thylakoid. Thus concentration gradient is created across the thylakoid membrane, and the enzyme ATP synthase is present in the thylakoid membrane that consists of two subunits, namely F0 or F0 and F1. The F0 subunit is involved in the proton transportation across the membrane, and F1, which protrudes on the outer surface of thylakoid membrane, faces the stroma. Abundant energy is produced due to breakage of concentration gradient. Thus their conformational change occurs in the F1 subunit, by which in light reaction plant produces ATP. We can write the summary of light reaction as 2H2O plus 2NADP plus 3 ATP plus 3 Pi are converted into 2 NADPH, 3 ATP molecule, and oxygen is liberated out of the cell. So we all understood that after the light reaction, ATP and NADPH are produced. But what is the use of ATP and NADPH? That we will learn next Friday while lining the dark reaction. Okay? So if you find this video helpful, please like and share it. And if you are new here, please subscribe and be a part of our Bioholic family. And today's self-assessment test is again in the description box. Participate in it. Now I am signing off. See you in the next Friday. Thanks for watching. Bye-bye.